Hello, my name is Leslie Domier. I'm a research plant pathologist with the USDA ARS. My lab is in the Crop Sciences Department at the University of Illinois. Today I'm going to tell you about some research we've been doing that is funded by the National Institute for Food and Agriculture on developing genetic nematicides for plant parasitic nematodes. First, I'll give you a little background. Um, First, plant parasitic nematodes cause billions of dollars in crop losses annually. One of the most famous plant parasitic nematodes is soybean cyst nematode that's pictured up here on the top. And on the bottom is a soybean field where you can see some of the damage caused by high nematode populations. Now, the best way and the way that's currently used to control most nematode diseases in plants is the planting of nematode-resistant crops. However, many nematodes, like soybean cyst nematode, exist as highly diverse populations, and they can, if the same crop variety or the same resistance gene is repeatedly planted in an area, they can develop the ability to grow on those resistant varieties by selecting out rare individuals from the population that now become dominant. As a consequence of we're looking to develop methods for nematode control that are independent of the plant host genotype and something that um, will not be influenced by the genetic diversity that is present in nematode populations. So the long-term goal of our research is to develop gene drives for the control of nematode populations. And those are techniques that have been used to control invasive insect species and also to provide some control for vectors of human diseases. First, I'll give you a little more background on how modified genes are transmitted through different populations. Normally, if we mutate a gene in a nematode, we will change just one copy on one chromosome of of the nematode. So that, and what we're proposing to do is to change genes that are involved in sex determination, so that a nematode with the mutant gene will most of the time develop into a male. So that if we can increase the number of males in a population to a very large extent, there'll be fewer females to produce offspring, and that will reduce the size of nematode populations. But in a normal genetic system, when we put a mutant gene out in the field, and that male nematode mates with a female wild-type nematode, about half the nematodes will have the mutation and will be male, and the other half will be wild type and can be either male or female. And then the same thing happens at the next generation. When this male mutant mates with a female, half of those progeny are likely to have the mutation, and half won't. And of course, all the wild type will then continue to be normal and wild type. And the same thing happens in the next generation. But what you can see then is that if we put out a mutation into a normal population, um, what we see is that that mutation is rapidly diluted and lost, and would have very little impact on nematode reproduction. But with a gene drive, we take advantage of the ability of the CRISPR-Cas9 system for gene editing to modify genes in nematodes or in other organisms. So we start off with something very similar. We have one mutant copy of a gene, and then on the other chromosome is wild type without the mutation. But what CRISPR-Cas9 does is it cleaves or cuts the gene in the wild type chromosome, and then the host repairs that, and what we end up with then are two copies of the mutant gene, and we still have male nematodes. But what happens then in subsequent generations is the same cleavage and repair mechanism goes on, so that when we mate the mutant male with a female, we end up with nearly 100% of the offspring being male. And the same thing happens at the next generation. When our male nematodes mate with a female nematode, they produce primarily male offspring. And then and the same thing at the next generation. So you can see that if we were to release a mutant male nematodes with this gene drive into a population, we could greatly increase the proportion of the population that's male and therefore reduce the number of females capable of producing progeny. So to do this, we need to develop systems to express genes in nematodes. So we're working on two different strategies. We have vectors for DNA-mediated transformation that's illustrated by the circular plasmid up here with antibiotic mixer markers and selectable markers and promoters and genes. And then we're also working with virus vectors where we're attempting to take a virus that infects nematodes and use that to express genes in nematodes. And then we need methods to deliver or to get these genes through the cuticles on the that tough coating on the outside of nematodes and into nematode cells. So we're trying a couple different techniques there. The first one is particle bombardment, using a gene gun to force small gold particles through the cuticle and into the cells. And the other method is electroporation, which causes destabilization of membranes and allows foreign molecules to leak into the nematode bodies. So for the first, for the virus vector system, we are using a virus that we discovered recently in sugar beet cyst nematode, a close relative of soybean cyst nematode. And we've named the virus sugar beet cyst nematode virus 1. And this virus is very closely related to a group of other viruses that infect invertebrates. And because those viruses are well characterized, we can use that information to help us or to guide our manipulations of this nematode infecting virus. So that we've identified regions of the virus that contain the structural proteins that form the virus particles and those that are required for replication. These groups of viruses tend to be very stable in the environment and also tend to be highly infectious. So we think it's a good candidate as a vector system for spreading genes to other nematodes. To deliver the virus into nematodes, what we've done is we've made a cloned copy of the virus genome, and then we take that cloned nucleic acid and we coat it onto very small gold particles. And then we use this gene gun to accelerate those gold particles and shoot them into the bodies of juvenile nematodes. And this slide shows some of the outcomes of that. On the first panel here is what happens if you shoot a nematode with too many gold particles. So this is a nematode really loaded with gold particles, and that killed the nematode. So what we did then was reduce the number and the size of the gold particles that we're using, and to a point where we have just a few gold particles in each nematode. And then the nematodes survive, infect plants, and produce offspring. So the strategy that we used was to collect eggs and cysts to allow the eggs to hatch, and then to take the second stage juveniles that emerge from the eggs and bombard those with the virus nucleic acids. We then allowed those juveniles to infect plants and then mature, and then about 4 weeks later, when the females had matured into egg sacs and the males had matured and climbed out of the roots, we collected the males and then assayed them for virus infection. And for the assays for virus infection, we used reagents and techniques very similar to those that are currently being used to detect the novel coronavirus that's causing COVID-19. So actually the reagents to do these tests are now difficult to get because they're so needed for detecting the coronavirus. So we took the RNA from the virus, from the nematodes, and put them into the real-time PCR machine, and along with primers and probes. And what we found was that we could detect, and that's shown by this blue line here coming up through the green line, um, significant levels of virus replication in those adult male soybean cyst nematodes. The negative controls, we saw no evidence of virus replication. So this suggested to us that we could inoculate nematodes with this virus, clone copies of the virus using gold particles, um, and produce successful replication of the virus. And we also wanted to see if we could express specific genes in nematodes. And the gene we chose was a luciferase gene from a deep sea shrimp. And the shrimp is pictured over here on this side of the slide. And these shrimp, as a defense mechanism, spit out a luminescent cloud that confuses prey that are going to eat them. And this luminescent cloud is generated by a small luciferase enzyme. So what we did was we took the publicly available sequence for that enzyme and resynthesized it so that it would look like a nematode gene. And that's what this cartoon is supposed to represent. This is our nematode version of this nanoluc gene from the deep sea shrimp. And then we use an electroporation apparatus to introduce that into nematodes so that we have a cuvette with electrodes, and then in the cuvette we have nematodes and copies of the gene, and we connect the circuit and we pulse the nematodes. And when we do that, this little cartoon illustrates what happens. Before we pulse the cells, the copies of the gene are outside the cell. During the pulse, the high electric current opens up pores in the cell, and some of the small copies of the gene go into the cell, and then once in the cell, those copies of the gene are expressed, and we detect those as luciferase activity. And we see during the electroporation, before we electroporate the nematodes, they're swimming around normal, motile. Immediately after we electroporate them, they're paralyzed for a few hours. And then after a few hours, they recover and swim around and act normal. And then when we assay those nematodes for fluorescence, what we see are very high levels of fluorescence compared to the negative control. And the fluorescence remains high for 72 hours. So again, what we've shown is that we can take a foreign gene and introduce it into nematodes and see high levels of expression of that gene. And we've done this now with 3 different nematode species: the root knot nematode, soybean cyst nematode, and also C. elegans, Cinereptitis elegans, a commonly used research nematode. So conclusions and next steps. So far we have successfully inoculated nematodes with a cloned virus genome. We've optimized conditions to deliver and express foreign genes in plant parasitic nematodes. What we plan to do next is to modify the genome of the virus to express inserted sequences. And this little cartoon down here shows a copy of the virus genome with a green fluorescent protein coding sequence inserted between the structural and non-structural proteins. And we also, now that we can get nucleic acids into nematodes and see expression of those, what we'd like to work on is being able to transform nematodes with genes and then have those transformed genes be transmitted from one generation to the next. This project has been a collaborative effort. Most of the research that I've described was done by a postdoc in my lab, Thanuja Thekividal, Chris Lambert is also a member of the crop sciences department. Chris is an expert nematologist and molecular biologist working on the project. And then the last collaborator on the project is Reza Hajimorad. He is a plant virologist at the University of Tennessee, and it was his lab who discovered the soybean cyst, sugar beet cyst nematode virus that we've been using for these experiments. Thank you for listening, and have a good day.